Hello dear, hope you are all fine and happy new year. Today we are going to correct the biology exam, the first term exam in biology for grade 8. Let's start with the first question, which is about the digestion of food. Number 1, we have to read the following text and then answer the questions. Please pay attention to the reading. Olive oil is rich in fats that are made of many molecules of fatty acids and glycerol. The digestion of fats takes place in the small intestine by the enzyme lipase and in the presence of bile. Bile is the juice secreted by the liver. What is the role of bile? Bile facilitates means makes easier the digestion of fats by emulsifying them, while lipase favors the chemical transformation of fats. At the end of digestion of fats, fatty acids and glycerol leave the small intestine into blood through the lymph. Now we have to pick up from the text these 4 questions. Question number 1. Pick up from the text the statement or sentence that shows the constituents of olive oil. Here it is. Olive oil is rich in fats that are made of molecules of fatty acids and glycerol. Also, we have to pick up from the text the name of the enzyme and catalyst responsible for digestion of fats or lipids. The enzyme is lipase, while the catalyst or helper is the bile. It is an exocrine juice. The route of absorption of fatty acids and glycerol. It means when they leave the small intestine, where do they go and by what or through what? Fatty acids and glycerol leave the small intestine into blood through the lymph. Pick out the role of enzyme and the role of catalyst also from the text here. We can say that bile facilitates the digestion of fats by emulsifying them, while lipase favors the chemical transformation of fats. Part B. We have to explain the following statement. Enzyme like a key or is like a key that fits only one lock. What does this mean? Of course, an enzyme, as we know, is a substance that can act only on one substrate or one kind of nutrient. For example, lipase, the enzyme lipase acts only on lipids. It cannot act with proteins or carbohydrates or starch. So this is the meaning of the enzyme is like a key that fits only one lock. Let's move to number 2. The table below represents or shows the quantity of glucose and amino acids in blood entering and blood leaving the small intestine. Of course, as we know, the glucose comes or is the simplest form of carbohydrates. Amino acids also is the simplest form of many chains or 52 chains of proteins. The amount of glucose, we have to compare the quantities here. The amount of glucose in blood entering the small intestine was 1 gram per liter and becomes 1.3 grams per liter in blood leaving. So the amount of glucose and the amount of amino acid are greater than the amount of glucose and amino acids in blood entering the small intestine. So the amount of nutrients in blood leaving is greater than that blood entering. What does this mean? A certain process is done here. Can you name the physiological process responsible for the above modifications? Yes. In this case, absorption of nutrients takes place in the small intestine. So what? So what now? Okay, we can say that absorption of nutrients is done in the small intestine. So the amount of nutrients in blood leaving the small intestine is greater than blood entering. Part C, we have to indicate the organic food that is the origin of glucose and amino acids. As we said before, that amino acids, its origin is protein, and glucose, its origin is carbohydrates. Carbohydrates is the complex form, while the glucose is the simplest form, as we know, or we can say reducing sugar. Let's move now to the second exercise or second question about immunology or the immune system. Number 1. Donating blood by compatible type. Blood types, as you know, are 4. A, B, O, and AB. So blood types are very important when a blood transfusion is necessary. In a blood transfusion, a patient must receive a blood type compatible with his or her own blood type. If the blood types are not compatible, so red blood cells will clump together, making clots that can block the blood vessels and cause death. Use the above information to describe how blood vessels can be blocked. Of course, yes, when the blood types are not compatible, so blood vessels can be blocked. Based on your acquired knowledge, indicate the other markers of tissue compatibility between the donor and the recipient. As you know, the self-markers of the body are 2. Blood types or blood group or ABO system and HLA molecules. So, the blood groups or blood types are mentioned in the text, so that the other type or other marker is HLA molecules. HLA stands for human leukocyte antigen. Determine how is blood type of an individual be determined, or how can be taken. Yes, it can be taken or determined by a serum test. Part D. We have to complete the following. Blood group AB donates to what? Of course, only to AB, and receives from all. That's why AB is called the universal recipient, because it can receive from all other groups. Blood group O donates to all other groups and receives only from itself or from O. So, blood group O is known as the universal donor. Number 3. The markers of self, which is the LA system, HLA system. Look at this text. Grade 8 students like you pose the following problem. How can our bodies distinguish between self and non-self molecules? Because it is this sentence ends with a question mark, so this is the problem, the posed problem. This distinguish is based on what? Depends on what? Depends on the nature of the certain protein molecules called antigen. So antigens are certain surface protein molecules that are expressed or located. Where are they located? Where are they placed? On the membrane of all nucleated cells. Nucleated cells, sorry, they are the cells that have a nucleus. Not all cells have a nucleus, okay? The most important ones are the proteins of the human leukocyte antigen, which is HLA system. But nowadays, the HLA system is still in use, but mainly under the name of major histocompatibility complex, MHC. What is its function? It is to define the compatibility of tissues and organs among individuals and What are its properties? They are genetically determined and called markers because they represent a biological identity card unique for each individual. They are not the same in all individuals. Each one has its own identity card. We have to pick out only from the document, not from your own or base knowledge. No, only pick out from this document. The post problem. As we said before, the post problem is mentioned at the beginning of the text in the first line. How can our bodies distinguish between self and non-self molecules? The post problem should be ended with the question mark. If you wrote the sentence or the post problem without a question mark, no any mark can be given to you. What is the name of the special identity card of protein in the human body? Of course, it is the self markers or HLA system. Okay, you can write whatever you need. We have to complete the following table also from the text. Location. Location, it is the membrane. They are located on the membrane of all nucleated cells. Function, they define the compatibility of tissues and organs and the properties or characteristics that they have. They represent a biological identity card for each individual or unique for each individual. This is all about the biology test, and you have please to correct this test on your biology copybooks. Thank you.